My name is Shreya Rangaswamy, and this is my project. It is about the cellular mechanisms and effects of arsenic, and I was mentored by Darby Kozan. So, I am a rising sophomore in the state of Washington, and I chose this project because I'm very interested in the field of cellular biology, and I was curious to learn more about poisons or toxins and their effect on our body. So, the contents there's the introduction, and we go into a deep dive into arsenic and its effects, and the conclusion. So, just an overview, I wrote a literature review detailing the biological effects and the associated health implications of different poisons and toxins like mercury, arsenic, lead, and anthrax. Each poison exhibits a unique mode of action influencing specific cellular proteins and pathways. Such reactions can trigger diseases in the body and ultimately affect the health and well being of the individual. In this presentation, we will explore the different mechanisms of actions of the poison arsenic specifically in the cellular level. So, why should you care? The prevalence of poison today poses a significant threat to human health, as there have been more than 2 million poisoning cases in the US each year. The number increases every year. This study contributes to the broader understanding of poison's impact on human health. The detailed analysis of arsenic could enhance awareness among the public, and the knowledge can serve as a foundation for the development of preventive measures, treatment strategies, and environmental regulations to reduce the risks associated with these poisons. So, now just some basic information about arsenic. Arsenic is a deadly toxin that can affect almost all organ systems in the body as a poison and as well as a carcinogen, which means that it can cause cancer. It has also commonly been used as a weapon for murder and bioterrorism because many arsenic compounds look like white sugar. Arsenic can act as a tumor promoter and helps tumors grow larger by changing the expression of genes that are involved in cell growth, and this results in the rapid reproduction of cells. Though it isn't concluded how exactly arsenic causes cancer, it might be involved in disrupting DNA repair, DNA methylation, and causing abnormalities in the structure of chromosomes in the cell. Arsenic is known to disrupt various pathways in the cell, like ones involved in cell migration, cell adhesion, cell survival, and others. So, people can get infected by arsenic through inhalation, ingestion, or absorption. So, most of the arsenic that enters the body is absorbed through the small intestine because the pH in that area is optimal for arsenic absorption. The poison molecules then go through a process called hepatic biomethylation, after which they form acids called monomethylarsenic acid and dimethylarsenic acid, which can harm the body in many ways. After arsenic enters the body, it can result in either acute or chronic poisoning, depending on the degree of its effects on the body. So, here's a bit about arsenic's cellular mechanisms. So, arsenic triggers the enzyme NADPH, which is nicotinamide adenine dinucleotide phosphate, shown over here, to increase the production of reactive oxygen species known as ROS, like superoxides and hydrogen peroxide. These ROS can bind with nitric oxide to form peroxynitrite, which is known to cause many cellular symptoms like modulations of cell signaling and necrosis or apoptosis, which is cell death. And as shown here, biomethylation also occurs in the cell, which is the formation of methylated compounds of metals and nonmetals. So in this case, as you can see, iron compounds are formed in the cell, which can disrupt many processes that happen in the cell. So now let's talk about arsenic-induced diabetes. High concentrations of arsenic can induce type 2 diabetes in the individual by reducing the expression of a protein called PPARY, which reduces the responsiveness of insulin in the body. Arsenic interrupts the production of glucose and restricts the ATP dependent insulin secretion, by replacing a phosphate group from ATP and instead forms ADP arsenate. Arsenic also binds with disulfide groups of insulin, insulin receptors, and enzymes involved in glucose metabolism. Arsenic diminishes the expression of insulin mRNA, the gene that encodes insulin, and increases the expression of many stress mediators like NF-κB. Also, high levels of arsenic can cause hypoglycemia as it decreases the activity of the protein glucose 6 phosphatase in the liver and kidneys. Arsenic can also cause hyperglycemia by slowing down the insulin dependent glucose uptake. So, now let's talk about the neurotoxic effects of arsenic. Arsenic can cross the blood brain barrier freely, and it also affects the nervous system by increasing the reactive oxygen species, decreasing superoxide dismutase, which is an enzyme that catalyzes many chemical reactions in the cell, and it decreases glutathione levels. And that's a molecule that's involved in tissue repair, protein synthesis, and other functions in the body. Arsenic also causes changes in the functioning of neurotransmitters like monoamines, acetylcholine, and glutamate. Arsenic can cause axonal degeneration or the loss of communication between neurons by destabilizing the framework of the cytoskeleton. This can cause thiamine deficiency in the body, which leads to encephalopathy and other neuronal problems. Arsenic inhibits the NMDA receptors in the hippocampus of the brain, which detect neurotransmitters and play a crucial role in learning and memory. Ultimately, this results in cognitive dysfunction and neurobehavioral diseases. So, this is about the arsenic's effects on the cardiovascular system. Arsenic activates the expression of genes related to atherosclerosis, which is an inflammatory disease characterized by the thickening or hardening of arteries, and some genes include HO1, MCP1, and IL-6. It also does this by increasing the synthesis of inflammatory mediators, which are molecules part of the immune response that induce inflammation, an example of which is leukotriene E4. Additionally, arsenic stimulates the protein kinase C alpha, which after many chemical reactions leads to increased permeability of the endothelium. Arsenic causes vascular endothelial dysfunction and many other cardiac issues by lessening the activity of endothelial nitric oxide, eNOS, which is shown in the picture here, which plays a critical role in regulating and maintaining a healthy cardiovascular system. Arsenic brings about vasoconstriction or the narrowing of blood vessels, cardiomyopathy, and ischemic heart disease, which is a weakened heart due to reduced blood flow. So, in conclusion, this paper explores the complex details of the cellular effects and health implications of common toxins like anthrax, mercury, arsenic, and lead to protect public health and advance the knowledge of problems of poison exposure. The research includes various modes of entry of each toxin into the body, the specific organs they target, and the important cellular pathways that they disrupt. Each poison poses distinct challenges to the overall health and well being of the human affected. Thank you.